I'll finish this, then we'll probably take a break. Then I posted the slides for the introduction to pluripotent stem cells. I'm not going to go over all of those today. What I'd like to do is come back to some of those things on Thursday, but I do still want to discuss that first research paper on Thursday. And today I really do want to give you an introduction to some of the techniques that are used. Some of them you might be familiar with, some of you might not be. Okay, so, but we will discuss that today. And anything that I don't cover today with the slides on the introduction to pluripotent stem cells, we'll talk about probably after the discussion of the research paper on Thursday. Okay. So everybody remember where we were with respect to what's happening early on in development, just to remind you. Now, we're specifically going to be talking about some things that happen specifically here. See how there's a big difference between the 8-cell stage where you can see the individual cells? These are literally compacting cells. And there's some really interesting but really important aspects of how those cells start interacting with each other at this stage, the morula stage. Before this, remember, you can take one of these 8-cell blastomeres, or even at the 16-cell stage before compaction is started, and you don't have any problem with the embryo. It's real easy. There's not quite tight interactions between the cells. But the cells reach a certain size, and they compact. They have to compact, and some things are happening at the cellular and transcriptional level that are really interesting in order for this very important decision, the first lineage decision, to take place. Remember, all of these cells before this stage are identical. Well, for the most part, we now know at the level of sequencing all the RNAs in individual cells, there are what could be some important differences. But for our purposes for right now, all of these cells are identical, phenotypically, biologically, and most importantly, with respect to what cells they're the progenitors for. Individual cells at this stage are just as likely to form any of the other cells in the embryo, any of the other specialized cells postnatally. At this point, they've made a decision. The cells on the outside take a specific differentiation developmental path. The cells on the inside, it's these orange-colored cells make a second decision. So now we've got 3 cell lineages. These are not the primary germ layer that we'll talk about. If you've heard these terms or saw them on the slide, this is not endoderm, ectoderm, and mesoderm. That happens later on. This still has to happen, and all this is taking place before implantation. Okay, so I just want to show you this live video microscopy. Okay, these are actually human embryos in IVF clinic. Yes?
Could you go back to the last slide really quick? Okay, so for the first lineage decision, is what like you're saying is that they decide to become a blastocyte and that's when they become less pluripotent?
No, we'll get into this. It's actually these orange cells here, not here. These darker orange cells make the decision between continuing to remain orange, and there's actually some molecular things happening that's not indicated here with the color pattern. But these cells make the decision between those cells and these cells. It's these cells, the orange ones, that's what's referred to as being the ICM, inner cell mass, sometimes called the epiblast. Those are the cells that will contribute to all of the cells of the embryo, and which, of course, contribute to all of the cells of the adult. They're fated to do that, the cell fate. That's what these lineage decisions mean.
And then the second one, specifically, like what does it mean?
I'll show you. Well, we'll go through that in a second. You'll learn the terms for both these and these. Yes?
How is the decision made? Is it random?
No, it's not random. That's why I want you to learn it, because it's just a beautiful example of how it's really complex interaction between the environment Meaning, at this point, they're just kind of floating along, not compacted, but they are probably receiving some signals. They're getting signals from each other cells, okay? And it's actually, you'll see the mechanism, and it all gets back to some of the events that are set up here during compaction. And you'll see some things, and they're unexpected. And I think this is a good kind of general, a good example of general aspects of how decisions that relate to cell function, directly relate to cell function, are made. Okay, and it involves external factors, it involves signaling molecules, we won't talk about those, cell-to-cell interaction and signals coming from the cell membrane to things in the cytoplasm, and then that signal gets transmitted to the nucleus to affect gene expression and transcriptional regulators, all kinds of things. You're just getting the tip of the iceberg, but I hope it intrigues you and makes you want to learn more. Okay, so this is, gosh, I can't believe this paper is 9 years old by now. But what they did, these are each an individual zygote. So however many there are here, and they're just being monitored through the microscope. Remember, these are all in the culture in the IVF clinic, just looking at it through the microscope. These were all fertilized at essentially the same time, but you can already see there's a big difference in timing of events, especially those cleavage stages. Remember, the cleavage stages are just multiple rounds of mitosis. But now you can start to see, now this one is starting to compact. Now, it's just shifted the microscope view on the stage. But now I think this one, one of these, is an incredible thing that's going to happen. They're just dividing away. Oh, now you're starting to get a little, there we go, forming that cavity. The first lineage decision has been made by this one and this one. See this little clump of cells? That's the ICM. That's what's separate lineage from the cells that are on the outside. And now this even more amazing thing is going to take place. It's growing, it's growing. Remember, this is in liquid. They're not really attached to anything. Oh, look at that. The blastocyst has hatched. You, at one point in your life, at this stage of your life, you hatched. Did you know that you hatched? Usually we think of hatching, chicken eggs hatching. You hatched. That's where that embryo, the blastocyst, breaks through those cells and membranes, and this is really kind of left over from the zona pellucida. Some of that is removed, and now I'm fuzzy about the details, but it's got to break through that, and it's not going to do it until that blastocyst reaches a certain stage. If hatching doesn't take place, that blastocyst is not going to be able to implant. So that's kind of, along with all of those cellular things and molecular things that are taking place with respect to lineage commitment, this is one of the key factors with regard to a successful implantation. Okay? No hatching, no implantation, nothing more happens with the embryo.
So they were all fertilized around the same time, and I understand why some may have thought I looks like there's only about 3 cells, but why don't they move at a similar rate?
That's why the study was done,
Okay.
to try to get some idea. And this is actually, remember I mentioned this I think last week and referred to it in passing today. This is actually the study in 2010. It's an early study. They actually, with other embryos, but under equivalent conditions, they're the ones that did RNA-seq. So sequencing of all the RNAs in individual cells. And they found some fundamental differences. And some of those differences, then they could, now of course they can't do RNA-seq on individual cells here if they're following it, but what they could do is they could relate some of the morphological differences, like these cells that failed past the first division, and relate that presumably to some key events that could have been happening at the level of sequence, RNA sequences. Yeah. Now that's that's $1 million question.
Yeah.
That's what they want to know. Because if they could find that out, perhaps they could find a way of, maybe, and again, remember, these are culture conditions in the lab. We hope that they're approximating as much as possible what happens inside the woman in the uterus, right? But there's virtually no way of knowing how close or how dissimilar they are. So, yeah. But at least with IVF, you could, if you learn this, you could apply it to the culture conditions. Yes.
Is there any like fitness advantage inferred from being the first one to reach that stage?
That's a good question. Now, let's see. I'm trying to think of what I know about twins, which is very little, either monozygotic or dizygotic. I don't know if they even implant at the same time, whether they're monozygotic or dizygotic. That's a good question. Now, yeah, these don't have much fitness, the really slow ones. I would think humans don't have more than one implantation the vast majority of times, other than with twins. I would think there would be something in the uterus that once implantation has taken place, it would signal that there wouldn't be any others.
But there's no like competitive or
I don't
like
know.
the equal to like those fastest.
That's a good
running-
question. Why don't you look it up? Yes. One more question.
Is there any difference in this process in aneuploidy cells as opposed to?
Well, yeah, that's a good question too. I'm trying to remember in that 2010 paper whether they could relate any of the RNA sequence analysis or even some of these morphological events to any aneuploidy, because you're exactly right, that aneuploidy could be taking place here. So this very well could be a mosaic aneuploid. That's a good question, That would, I would think, in most cases, that would have to have a profound effect on the embryo. But okay, then maybe not, because remember at this stage, I mean, if this were to continue to divide, if the aneuploidy resulted in lethality of one or two individual cells, the rest would continue on. So maybe not. Good questions, excellent questions. I like them. Okay, take a look at those on your own. You should be able to click on that video. Not if you just are displaying the slides in ELC, you'd have to download them to be able to see that video and any of the others. Okay, so this is what I've been referring to. We've got the blastomeres, so relate the terms up here and the events taking place to what we have down here. These are similar drawings of the cells. Blastomeres, individual, equal potency. Now, the 16-cell morulates, this is where it's between the 8-cell and 16-cell stage where the cells are starting to compact. But already you can see something happening here. We haven't had the first lineage decision just yet. But see how you can see this ring of outer cells with cells on the interior. Here, they're all exposed to the exterior. There aren't any on the interior. Here there are. That sets in motion at this point forward. These cells in the outer are having molecular and phenotypic cellular differences compared to the cells on the inner. Okay. Now we've got the first lineage decision has been determined. ICM, inner cell mass, okay, and TE, trophectoderm. So ICM, trophectoderm are the result of the first lineage decision. And it's all set in motion because of this outer and inner organization of the cells. Then the second lineage decision that takes place, the trophectoderm continues on, and these cells will contribute to the placenta, the embryonic portions of the placenta, and some other embryonic derived tissues. The TE, the trophectoderm, will not, under normal conditions, contribute to anything having to do with the embryo itself. It's essential for the embryo, right? Because if you don't have the placenta, the embryo is going to most likely die. Or will die. Okay. Then the second lineage decision takes place with the ICM, forming these two terms, epiblast, and you can call this hypoblast if you want. I prefer the term primitive endoderm, because we'll see how the primitive endoderm later on develops into the definitive endoderm. We'll see more about that in a second. So I do want you to know all of these terms, the first and second lineage decisions. Okay? And just to show you some beautiful pictures, this is actually out of one of the textbooks. I think this is chapter 3 of Carlson. I think that's a supplementary. No, I forgot. 2 cell, 4 cell, 12 cell. Look at the difference here. That's compaction. You can't identify individual cells at this point. Now, if you were to cut into that or do either physically or through microscopy, taking optical sections, you would be able to see cells on the outside that are oriented differently and have different properties than cells on the inside. There we're seeing this. The morula and the late. Now, not only do we have these outer versus inner cells, we're starting to get some fluid that makes up that blastocele cavity. This is like blowing up a balloon with water or something. This is not air, this is fluid. This fluid is really important. Here's our TE, the trophectoderm, there's the ICM. Then here's the hatching blastocyst. Okay, so before we even go any further with the details, why is this interesting? Why do I think it's interesting? Why do I hope you're interested to you? Why do I want you to learn it? Yes?
So, if you're looking on the blastocyst on the screen, for example, do you see it flat because it's a cross-section because of the microscope, or is it flat because it's developing on a flat surface?
Okay, that's a good question. These are essentially balls, circles of cells. These are not attached to anything. I think that's a good point, because other types of cells that we'll talk about, and I think I probably mentioned, will attach to the bottom of the dish and be flattened out. These appear to be flat only because anytime you've got a microscope, the traditional kind of microscope, you can only see one focal area of focus. Now there are, maybe some of you know about these laser confocal microscopes, where you can actually take multiple optical sections and go through and reconstruct a 3D rendering of the embryo. But yeah, these would be circular. Yeah, so think about those embryos in the video. They're kind of moving around, and you can see they're kind of rotating around. Yeah. Okay. Fundamental questions. I mean, cell fate specification happens virtually everywhere in the embryo. To a certain extent, it continues to happen even postnatally. Not with every cell type, of course, but it has to happen to make a liver cell different from a brain cell. Okay? But more, even going beyond that, we can use these pre-implantation embryos to determine totipotency versus pluripotency. Pluripotency and totipotency are terms that you'll learn when we talk more about stem cells. Pluripotency is the ability to form all the embryonic lineages. Now, what's important to realize is that pluripotent, this term, leaves out an important type of cells. Pluripotent stem cells cannot contribute to the extra embryonic tissues, the placenta, the trophectoderm. Only totipotent cells can do that. And that's an important distinction that we'll come back to. In the embryos that we've looked at so far, only the morula cells would be considered to be totipotent. Pluripotent cells would be the ICM. They've already made that fate decision between trophectoderm and the inner cell mass. Okay, so that's why they're not pluripotent. And this is where we're getting, how do those cells that are all identical at 8 stages make the 2 lineage decisions? Their fate is determined. And remember, these are all genetically identical cells. Individual epiblasts cells should, with the exception of aneuploidy or, mutations or other mistakes that take place in mitosis or after, they should be genetically identical. So what makes the cells in the epiblast destined to become all the cells in the fetus, to be able to differentiate into all the differentiated cells, and those that develop that way in the fetus, of course, they're programmed that way to become all the cells postnatally. What makes TE cells only destined to the placenta? Now, this is why I prefer the term primitive endoderm. During this special stage of embryonic development called gastrulation, it takes place after implantation, so we're not going to consider the details of it. But there's a really cool thing that happens with the PE that converts it to the definitive endoderm. But we'll see the importance of that in a minute. And this is the million-dollar question with respect to pluripotent stem cells. How do these retain their pluripotency? And we'll look at some of the mechanisms. That's what the paper that we'll discuss on Thursday. It doesn't get at all the mechanisms, of course, but it just gives you some idea of how they obtained information about several really important mechanisms. Clinical importance. I mean, this is the crucial stage in human embryonic development. Only a third of them make it. And half of that 1/3 is pre-implantation. And I've already mentioned this. If we can understand, well, it kind of goes both ways. Not only are pre-implantation embryos really important tools for stem cell research, stem cells are really important tools for understanding pre-implantation development. Now, as we'll see, you won't have to learn the details of the species differences, but as we'll see, there's a lot of things that are very similar between mouse and human. And so many aspects of mouse embryonic stem cells and mouse pre-implantation embryos should apply to humans, but we already know there are some fundamental differences. So given, the difficulty of working with getting human embryos, both in a practical sense, in the ethical, political, whatever, all of that sense, it's hard to get human embryos to do research on. It's easy if you're a mouse geneticist or working at a university or other research institution and mice, you can get lots of mice. Scientists would always know that there could be some fundamental differences, fundamental similarities. So you take advantage of both. Okay, so this is animation, it's not live. I recorded it from that textbook. There's no audio. I'll just provide the audio. Here we've got a zygote tumbling along in the oviduct. Just heading, that's the first mitosis, blastomere, zona pellucida on the outside, remember? Then it's just kind of tumbling along. Now this is where it is asymmetrical. You do get a 3-cell first and then a 4-cell. But after the 4-cell stage, again, the blastomeres are identical. They're still enclosed in the zona. 8-cell is coming up. Oh yeah, remember here's where the zygotic or embryonic gene expression begins. Now we don't have any difference. This is a little bit misleading. There's not inside-outside cells just yet. Now this is all in the fallopian tube. Now we're reaching there's that compaction. We'll look at this in more detail. The cells have become different. There are outer cells and there are inner cells. First lineage decision, and shortly after, that cavitation, which is the formation of that blastocyst cavity, fluid-filled. There's ICM, trophectoderm, or trophoblast. Individual cells in the trophectoderm are called trophoblasts. Then it hatched. Now it's separate from the zona now. It no longer needs the zona. Now it can implant. And it's tumbling along. I don't know what it's looking for. I don't know if this is completely random or not. One thing to mention, there are some really important cell-cell interactions. Let me just pause this. Some real important cell-cell interactions that take place between the embryo, particularly the trophectoderm. Now this is the lining of the uterus. And what we're gonna see happen is that these cells will literally invade through, continue through, and move through the uterine epithelial cells. That's what these are. Now remember, the uterus has been primed by hormones. Okay, so this wouldn't be like the typical uterus. This is just right after ovulation. The uterus gets a surge of, I've forgotten all the different hormones, but that makes all kinds of things take place in the uterine lining, in the epithelial cells here. So this is the interior of the uterus. This is the wall of the uterus. And not only are there changes in these epithelial cells, there's all kinds of changes taking place in the non-epithelial cells. Okay? And this kind of invasion of the blastocysts, in many ways, this is very similar to what happens with cancer cells that are locally invasive, right? Because cancer cell has to break through the normal boundaries between tissues before it can get into the bloodstream or the lymph system to be able to seed, travel to other tissues and metastasize. So some people actually study this process as a way of understanding its relationship to cancer cell invasion. Okay, now you don't have to know all the details of this, but just know the second lineage decision has taken place. You don't have to know these terms. Okay, there's our invasion. Now there's all kinds of signals going to the blood vessels and there's all kinds of things that are happening in the uterus as well as in the embryo. What's the other term for hypoblast?
PE.
Primitive endoderm, PE, yeah. And other than the growth here, for a while, not much is really happening within these cells. Some things are happening in the trophoblast, there's things happening with the uterine cells, and again, you don't have to know all of these terms. It just keeps growing. Then we're gonna get this stalk growing down. Now this is presumably human. I think at this point there might be some differences between mouse and human. I'm not sure about that. Oh, we'll see that later. And again, you can watch that on your own. Okay, so I just wanted to show you this. So on the bottom panel are all the things happening up to the blastocyst, the late blastocyst in humans. Now E, both with human and with mouse, refers to embryonic day 5.5. So 5.5 days essentially after fertilization. Okay, so that gives you the timing. Virtually identical cellular events taking place in human versus mouse, but the timing is slightly delayed in humans. Okay. Now, if you took any placental mammal, rabbit, dog, cow, pig, it'd be essentially the same. Again, timing would be different, but essentially the same thing would be taking place in any placental mammal. Now, there is a little bit of a difference here. You notice here in the human late blastocyst, we don't have that third layer of cells. That actually takes place slightly after implantation. And even though I told you that essentially the same processes, just slightly delayed, are taking place in human and mouse, some of the details are more complicated in human. So just because it's simpler to explain, I'm going to explain some of the details, not all of them, some of the details taking place in the mouse. But I want you to learn them as examples. Just know that not exactly the same thing is taking place and at the same time as in humans. Okay, so that's where we're taking advantage of mouse. Relatively good model. Okay. So again, just to orient you, here's the cleavage stages, but now look at what's happening here. We're still getting mitosis. We're still getting these cells dividing, but they're asymmetric. We'll see what that means in a second. But here's the timing. There's our first lineage decision. This, roughly, in here is where compaction takes place. We go from that, which is not compacted, but it's setting up the events for compaction, to here, where we've got the inner and outer. inside-outside positioning. That is critical. Then we get the blastocysts and other things that are taking place. We'll look at just a little bit of them. I'll mention just a little bit. Things that take place here set up the ability for fluid to be pumped in to the interior to form the blastocysts. And again, if you don't form the blastocysts, you don't get the implantation. Yes?
You're going from the 8 cell to the 16 cell. Is it the same cells that make up, like when it divides? Does it go from with the 2 daughter cells, one be inner and one be outer?
That's still being investigated. I'll show you a little bit about that. I know more of what, and I'll show you what's happening here. I don't know the details of that, but that's a good question. I would suspect if somebody hasn't found the answer to that, they're still looking for the answer. Yeah, but no, that's a really good question. Okay, so what's happening with this compaction? So if it doesn't compact, you don't get viable embryos. You don't get that first lineage decision. You don't get blastocysts. You don't get hatching. Now, anybody ever heard of adhesion molecules or E-cadherin? Yeah, you have, like in a cell biology class, or where have you heard of it?
They tested for the
Oh, okay, MCAT. Had you studied it? Did you know what it had been? Did you learn it in a class here, or refer here of it in a class here? Yeah. So you don't have to worry about the details, but it is a protein. It's specifically involved in making these connections between cells, between adjacent cells. And don't think of it as just being the static structure. I mean, in a sense it is, but it also is really important for conveying signals that cells are attached to each other, which is an important thing, since because of that attachment, there are signals that are being transmitted through the cytoplasm into the nucleus to affect gene expression. Now this term polarized, we'll see an example of this. Polarization of cells, or cells becoming polarized, doesn't happen just in the embryo. All epithelial cells, like we saw in the uterus, the lumina, the uterus, okay, those are epithelial and they're polarized. All the cells that are on the interior of your intestine are polarized. They have adjacent cells that they make connections to, and one surface of the cell is exposed not to another cell, but to fluid or connected to just something. Okay, so that polarization is really important. And that doesn't happen unless E-cadherin changes where it's located in the cell, and both of these play an important role in compaction. Now, you don't have to know the details of it, and these aren't pretty color images, but what's lightly staining here is fluorescence, just grayscale. So the light areas show you where E-cadherin is localizing. You see a difference between like this cell and this cell, or even between these two cells, or between these two cells? Do you see anything different? Don't just read what I've got here. Look at the images.
You're talking about like how they're like right where they're touching. It's like they're one.
Here, now we're not looking at the nucleus. This is the outside of the cell, so we don't even see the nucleus. It's just the outside of the cell. It goes through a very dramatic change in localization. At this stage and this stage, it's essentially all over the cell. Well, in the cytoplasm of the cell. At this stage, see, this is where those junctions are forming. But also, we'll see it more, and it's more pronounced here. These are the outside cells. Can't see a real one here, but they would be inside cells here. These tight connections between adjacent cells here and here are taking place in the cells that will become only the outer cells. This is a blow-up, a diagram of this polarized cell, 8 cell, it's starting to compact. But now look at here, the color coding, and I'd like you to know these terms. So this is an individual cell, right here it's that cell, and then the cell to the left and cell to the right, you just see parts of that. The basolateral domain, basal, so if you're thinking of this being the outer, apical, apical kind of at the top versus basal. But basolateral, meaning towards the bottom, but to the side, basolateral. That's where incoherent is making those junctions. This is there's other things, other proteins there, but this is what's dramatically relocalizing between here and here. Oh, another thing I wanted to point out, you might be interested to know, that the bulk of the ecoherin protein that's made here has been translated from maternal mRNAs. And remember, zygotic, embryonic gene activation isn't taking place till here. It does turn on expression, the zygotic expression contributes to this, but probably most of this is probably a combination of maternal and embryonic. This stage and this stage I would suspect would be almost all. This is all maternal, this is probably a combination. Okay, so this is where they become polarized, by having the apical versus basolateral domains. That's what refers to being polarized. There's different cytoplasmic things, different things located at the cell membrane, by things I mean proteins, than here, okay? I don't know who was first. You go first, you're in front of them.
When we can hear the lessons like this box where there's the cell junctions, are they secreted out of the cell?
No, it's intracellular, it's only intracellular, as far as I know. And I'd be real surprised if any is extra. So, and this is all in the interior of the cells. Now, of course, there could be other extracellular proteins that have an extracellular domain that could be embedded into the cell membrane, but not ecoherent. Yes?
What's the significance, if any, of the number of cells that it gets to before it starts to hatch?
Well, I think the I don't know. Okay, the numbers are important. I don't know if it's the absolute numbers, but somewhere in the 8 cell to 16 cell stage where compaction starts, the numbers are important just because of this, and I don't know the details, but it's pretty clear that somehow the cell is sensing either the size and or the number of cells. Because it wouldn't compact if it was only at the 4 cell stage. Yeah. And I don't know if there has to be a certain number of cells at the ICM stage or a certain number of trophectoderm cells. Look, I know. I'm kind of being facetious, but if you're interested, I can't tell you everything. I wish I could. No. Okay. Now this is the other cool thing that you might not expect, but if you think about it, maybe you would. Now we've got compaction, fully compacted. We've got basal, basolateral, apical polarized cells. But look at these dashed red lines. And we're looking at mitosis in the outer cells. Now mitosis on the inner cells is taking place too. But mitosis and cell division in the outer cells. But it's kind of half of it. I think it's half. I don't think it's biased one way or another. But if the mitotic spindle in this progenitor, in this cell, if it lines up vertically like that, just imagine the 2 daughter cells going left and right. Those will stay on the outside. And you'll get 2 outside daughter cells. If the mitotic spindle lines up this way, look, you'll get 1 cell outside, 1 cell inside. But now this is also showing you something really important. Look at the blue versus the purple and this morula versus that morula. What do you see that's different with this inside cell and that outside cell? Remember, those are presumably identical daughter cells. Are both of them polarized? Why are you saying no?
Because the inside cell has only the basolateral domain as opposed to the basolateral.
I wouldn't even refer to it as basolateral.
It's
That's
touching
a-
cells on
yeah.
all sides.
Yes, it's receiving very different set of signals than this cell. Because it's only, this cell on the outside only makes contacts three cells, and it's got the apical domain that's exposed to the outside. Did you have, was there another question? You and then you can ask another question, yes. I'm sorry, I haven't learned very many names yet.
Is there any instance where a cell that divides and like starts to be molecular change to the part of the ICM is kind of pushed out because of another division and has to change? What could it wait for a little bit?
I don't know. That's a really good question too, making me feel like I should learn more details. This is great, no, and I don't have any problem admitting to you guys where I don't know things. But saying these are really good questions, yes, like you had one more.
Yes, so I was just wondering, based on that, the difference in signaling, would we then see that like certain cells divide more frequently than other cells? Like some cells might arrest their development?
No, I don't think any of them will be arresting in
Okay.
mitosis here.
Okay.
Because remember, they're gonna now make, perhaps, once this gets bigger, perhaps the trifectoderm cells.
Right. Okay.
Now, going back to your question, I think once that cell is committed to the ICM, it wouldn't be becoming trifectoderm. Is that what you were asking?
I was asking like if it was already committed to the ICM and then somehow it got pushed out because of another cell division onto to where it's exposed, would it change because of the result of cell signaling?
If probably, yeah, because if it say it was exposed, like once the blastocell cavity starts forming, and if there's a cell that by accident got extruded into the cavity, it probably died. Or just not go. That's where it might arrest. I don't know. Yeah. Okay. So we're continuing to have outside cells becoming more trifectoderm, inside cells becoming more cells of the ICM. Now this is where we start getting the formation of the blastocell cavity. Because once this takes place, it's only the outside cells, and I'm sorry, not here. Here, it's only the outside cells that are exposed to the exterior. They'll actually start pumping ions in and out, and that causes water. Remember, this is pre-implantation. It's not like, the uterus isn't completely fluid filled, but there's a lot of fluid there, right? And that's that osmotic pressure going inside the cell just forces those cells to start expanding, the trifectoderm. Now, there are 4 transcription factors that I'd like you to learn. These we'll come back and talk about several times with respect to pluripotent stem cells. These, in terms of what I want you to learn for this class, we're only going to be talking about these in the pre-implantation embryo. Now, I don't think this protein T4 plays the same role in human. That's one of the species differences. But again, remember, this is what I'm asking you to learn here is mouse. They're all transcription factors. These we'll see are important not just in the embryo, but as I said, in pluripotency. They can be both positive and negative regulators of transcription of other genes. T4 is responsible for activating expression of CDX2. CDX2 is transcribed only in the trifectoderm. I'm sorry, I shouldn't say that. It has different, no. it's acting as a transcriptional upregulator, a positive regulator in trifectoderm, and activating expression of genes required for trifectoderm function. It acts as a negative regulator in the ICM. Okay, now we've added this figure from another review article. You don't have to read it. Now this color coding will tell you that CDX2 has been turned on in the outside. OCT4, as we'll see, is a real important pluripotent master regulator, both with embryonic stem cells and, as we'll see, with induced pluripotent stem cells. Don't worry about GATA6. Okay. But look at the color coding here. Blue, green versus this red. There are different expression levels. So we do have different levels of expression of CDX2 in the inner versus outer cells. On the inner cells, low CDX2 will cause higher levels of expression of these other transcription factors. In the outer cells, which are affected from high CDX2, will cause low expression of all the other transcription factors. Don't worry about the difference between these two models. In fact, it's probably a combination of both of these. But there are very important, this is where the polarization of these cells comes into play. The high cell contact at the basolateral domains versus exposure to the outside and low cell contact at the apical domains of the outer cells. This is just giving you a diagram. This is just immunofluorescence staining for, always pay attention to the color, these aren't color-coded labels, but this is with green fluorescence. Now, anytime you see OCT3/4, just know that's the same as OCT4. In mouse, it's called OCT3/4, and human will see OCT4. It's the same thing. Here is the CDX2 expression in the outer and trifectoderm. Now you can merge digitally with a computer attached to the fluorescence microscope the green signal with the red signal. And this is taking advantage of the awesome power of mouse genetics. You can actually make knockout T4 mice. Look at these. You don't even have any CDX2 that's telling you it's a positive regulator of CDX2. Right? In the absence of T4, you don't get any expression of CDX2, but you also don't get that first lineage decision. All of these cells stay OCT4 positive. And DAPI is a staining blue, well, almost always you see it as a blue signal for DNA, the nucleus. Now, we're not going to talk about, so this is the example where CDX2 as a positive regulator for trifectoderm-specific genes, as a negative regulator of OCT4, which is required for ICM expression. Since we know that T4 is a positive regulator of CDX2, we should be immediately thinking, well, what activates T4 expression? That is a really, really cool story. But you're not going to have to learn it. But it does involve signals from the membrane. It involves a protein that gets phosphorylated. It involves a change in cytoplasmic location versus nuclear location of phosphorylated protein. And it involves a protein that's a co-activator for T4 to activate CDX2. Yes? Yeah.
I'd say co-activators.
Yeah.
I was going to ask why T4 didn't
Okay, I guess we should take a break now.